So, welcome to the next talk for the InDepth Academy sessions. So, as I know, these are part of the InDepth Academy, which is something that the InDepth Cost Action has instituted to bring together an online resource of talks and tutorials and also protocols to help the community. So, and as part of this, Dionysia will be giving a talk about her work on Maize. So thanks very much for joining us in rather warm Cyprus, as you said, and we'll look forward to hearing your talk. So please go ahead.
Thank you very much for the introduction, and it's very nice to be able to communicate with you again. Thanks to the organizer for giving us the opportunity to present our work with maize. From the Agricultural Research Institute in Cyprus, I'm going to talk about maize above ground and root microbiome phenotyping, exploiting novel field phenotyping equations. So plant breeding, I'm a plant breeder and molecular geneticist. So plant breeding is based on phenotype. Field phenotyping, as opposed to phenotyping under controlled conditions, presents multiple challenges, yet is the essence of plant breeding. Our approach is quite novel and in relevance with the strategic plan of SEB about cross-fertilization of ideas and disciplines and that have public benefits and impact. And for those of you that are interested, you can take a look at the recent report about the socioeconomic and environmental values of plant breeding in the EU and selected member states by Nolepa and Katzberg. Now, a critical question that we have to face, given the unique features of plants as opposed to animals, and these features include this immobile nature of plants, the root systems, for example, the multiple progenies that the plant produces as opposed to an animal. So given those, what should be the unit of plant phenotyping, including for plant breeding purposes? And when we say plant breeding purposes, we refer to the selection of the best plants in its environment so as to advance the most appropriate to the next generation. Now, the problem we face in plant breeding is that evaluation is based on multi-plant densely grown plots, but density suppresses phenotypic expression and differentiation. These are the classic density grown plots for maize and barley here. Now, we advocate that the appropriate unit of field plant phenotyping is the individual plant. But how? What kind of individual plant? When grown in conditions that maximize phenotypic expression, that is, in the absence of interplant interference, meaning no competitive interactions between plants. You can find more about this in this topic, in this presentation, where we discuss 7 common barriers faced in plant breeding programs, and we propose specific solutions. So to be able to face this challenge, it is important to define competition. We have defined competition as a plant-to-plant interference with the equal sharing of the density-limited growth resources. It can be caused by genetic and acquired differences, and is quantified by the CV, the coefficient of variation of single plant units. The definition of competition is very important, both because it provides a means to quantify it, and also because there is a great confusion between the stress from competition and the stress from density. So we say that competition is not equivalent to density stress. when we have or we create the right genotypes, it is possible to have high plant densities, the cropping stands, and at the same time have reduced interplant competition. Further points to note is that competition hinders the efficiency of selection because of the existing negative correlation between yielding and competitive ability. Now, more barriers to field phenotyping, including root phenotyping on site, that is, under real field conditions, involves the soil or spatial heterogeneity. This soil heterogeneity exists regardless of the soil microorganisms and refers to soil fertility or soil water content or soil structure and anything like that. But the biodiversity of soil microorganisms, including those tightly associated with plant roots like the AMFs, introduces additional heterogeneity. Now, people have recently started exploring studies of rhizobiome at the single plant level, and this is a very recent recognition that there are very few studies exploring spatial variation relevant to direct interactions between fungal species with their hosts within the root system of plant individuals. So in our approach, the wide interplant distances coupled with innovative field selection designs that are specifically created for field phenotyping and comparative evaluation of individual plants. Okay, so this is the same field design under two different views. The color map represents microbial diversity, where its color corresponds to a different AMF species, and it shows how this diversity can be effectively sampled when we have the appropriate designs. So this is one of the multiple honeycomb selection designs that is capable of evaluating 90 different entries or families or genotypes. So notice that in each row, this is field rows, and each number corresponds to an individual plant belonging to family number 19 or family 9 or family number 2, etc. And each plant has at least 1 meter distance, interplant distance, from its neighbors, so as to exclude interplant competitions. There are situations where we can use 2 meters distance from plant to plant, depending on the kind of species or the kind of plants we are dealing with. So in each row, there is an ascending order of numbers, and when this is finalized, in the field there are formed multiple complete and moving replications. By complete, we mean that each plant, regardless of where we look, it is in the center of a complete replication, meaning that the remaining 18 representatives from the 18 other families surround it. And the same is here in the other part of the field, when number 5 is at the center. And the same is the case with any plant at any point in the field. At the same time, representatives from the same family, for example, all number 10s, plants belonging to family number 10, form triangular moving grids that span the whole field for spatial heterogeneity, providing a really effective sampling of it. Now, the importance of it is also that once we have effective sampling of soil heterogeneity, we can safely ascribe differences in entry variances to genetic differences in stability of performance. Additional challenges of our times. In the past, in the paradigm of green revolution, it was based on crops responsive to high soil fertility, as fertilizers became readily available after the Second War, and ample water availability. Selection of plants under these conditions has affected the ability of modern cultivars to establish beneficial associations with soil functional microbial assemblages, like the AMFs or nitrogen-fixing bacteria. So the community structure of these microbial guilds is crucial for the productivity of agricultural systems. Here we see this concept in the graph. So under modern plant breeding, the plants adapted to agricultural practices of high fertilizers and use of multiple pesticides, tillage, etc. The AMF diversity is reduced. The AMF becomes less important for plants because they have all the other resources, and this leads to loss of the ability to form efficient symbiotic associations. In the future, the focus, and here it's already, we are dealing with this, the focus is on successfully selecting genotypes tolerant to biotic and abiotic stresses with high stability of performance and capable of exploring the beneficial associations with the soil microbiome. The AMFs are known to facilitate nutrient uptake and crop tolerance to abiotic and even biotic stresses. And this is a graph to depict AMF importance for plants and the different physiological functions and biochemical functions they are helping with. Also, in the future and now, the focus is on exploiting the substantial differences between the reproductive lineages of plants and animals, and this is highly relevant to the work done by the cost InDepth, as for example the work by Celia, Baruch, and others. And we need to be able to capture favorable epigenetic events or genetic events during plant development for breeding purposes. But for this, it is necessary to work on the single plant level and under the concepts I just presented. So for this particular series of experiments, here you see a view of our maize field where we have the individual maize plants allocated according to one honeycomb selection design. We worked with 7 land races and individual selections from those. Plants were excavated, roots were washed, collected, stained, preparation of slides, etc. This is the work of my colleague, Michalis Omiru. So, and this is a view of slide during the AMF colonization. And we see vesicles for fungal survival and maintenance in the maize unit. So all these structures have to be counted under the microscope. And we arrive at the so-called colonization index, colonization percentage. And when we plot this percentage against the yields, the individual plant yields from the field experiment I just showed you, we get a series of graphs. And this tells us that colonization is associated with yield, but this is genotype dependent. And, for example, each dot here represents individual plant from the landrace Abelanto. Same, each dot here, individual plants from the landrace Lavec. And the same for the other landraces. You can see how easy it is to isolate individual plants with contrasting behavior in the field under real field conditions, and the significance of this for molecular studies or biochemical studies or physiological studies. So then AMF diversity was further assessed through PCR DDG analysis. A cluster analysis of maize mycorrhizal interactions showed that it is possible to combine microbial AMF diversity with high yields, but in some cases, higher yields are associated with lower diversity. So to summarize conclusions, phenotyping at the level of single plant growing in the field in the absence of interplant interference using the honeycomb selection designs can bridge the gap between the epigenomic and field analysis, yield data. There are multiple novel opportunities for plant breeding and plant biology. It's a powerful tool for evaluation and selection of plant genotypes that possess the capacity to form beneficial associations with soil microbiome, including under the fluctuations of climate change. It is possible to perform direct and reliable selection for yield under real field conditions, as opposed to using various physiological traits or biochemical as proxies for yield. And this approach resolves also automation challenges in planting. And more specific, different maize genotypes exploit differently the native microbial communities. Striking effect of genotype on AMF colonization and AMF diversity shows that it is a validated target for breeding purposes. The increased colonization is associated with biomass production and yields, but this is genotype dependent. And the ability to select for stability or homeostasis at the single plant level leads to creation of density neutral or density independent cultivars. These kind of cultivars are very valuable under climate change because the crop performance does not depend on the density of the stand. In other words, we can achieve high yields with less seeds per soil unit. And this can happen also when we have drought or other events that destroy some of the plants during the cropping season. So I believe this is our last slide. Thank you very much for your attention. And this is our colleagues working on this project. And if I can take any questions.
Thanks very much. Thanks very much. That was very interesting. I remember discussing the honeycomb plot with you previously at I think a meeting in Prague, wasn't it? It's
yes.
kind of fascinating quite a fascinating system to do that. So I have a couple of questions. So first of all, the honeycomb, just to get it clear in my mind, it's so each number is a particular genotype or a particular land race. So it's individuals from the same genotype are spread at different areas. So I understand that you're seeing different soil interactions, but within that, each plant is next to the same plant at each position. So if you're getting any interactions between genotypes, then this wouldn't rule that out, I'm guessing. Is that right?
I'm not sure if... Okay, I'll start saying something, but if I don't cover you, please ask again.
Mm-hmm.
Its number represents a single plant, but this is in at least 1 meter distance from the neighbors. So there is no plant-to-plant interference.
Okay.
However, okay, so here, plant that belongs to the family number 1, okay, or to the variety number 1.
Mm-hmm.
Here we see it is positioned in a field spot where the red species of AMFs dominate. But here the same plant, for example, the same genotype, is positioned in another spot of the field where another microbial species is dominant. And here is yet a totally different situation where the same plant, it is positioned the same, how to say, not the same plant, but the same genotype is positioned in a completely different field spot. So the number of replications when you work with individual plants is big. So it's always above 30 easily, and you can use even 100 replicates or even more per family or per line. And all these are allocated in a way that samples spatial heterogeneity very precise. And also regarding the neighbors, here, this is the same design, okay, but for example, number 1 here, it's number 1 here. But let's work with number 10 here, which is this number 10, okay. So here, number 10 is surrounded by representatives from the remaining 18 families. 1, 2, 3, 4, 5, 6, 7, 8, 9, 11, 12, up to 18. So this is complete replicate. But this kind of complete replicate forms each plant in every spot in the field. So number 11, the neighboring plant, possesses again a full, complete circular replicate from all the other representatives surrounding it. And this is the case in every spot in the field. Here, number 5. It's surrounded by representatives by all the remaining. So, okay, where is the equation? Yeah, thanks, Gary, because I didn't say that. So, the yield of each plant, here, the yield of plant number 5, for example, is expressed as a function against the mean yield of all the remaining plants in this yellow circle. And it is multiplied by a function that corresponds to the stability of performance. And this simple measurement gives a value of a plant phenotyping equation. So each plant in the field has a very specific field position that we are able to work with methods of georeferencing, and next to its very specific field position, it can have the value of its phenotyping equation. And this value, of course, is different from field to field, but it describes the situation perfectly, the situation in each particular plot, in each particular field.
Yeah, I see. So, I mean, it really is a marvelous system for maintaining variation across the field. So, I've got a question now about the results that you had. So, that nice diagram and the nice graph you showed with the different colored spots. So, you showed that some of the lines, some of the genotypes had increased yield with increased diversity, whereas others had increased yield but with less diversity. So was that unexpected, and what does that mean, do you think, in real terms? It's just the specific functions of the genotype, I guess, are better adapted, or what's your interpretation of that?
It was not really exactly unexpected or expected, because, as I mentioned, it is a very novel field to start working under real field conditions with this kind of approach. Okay, so we didn't really know what to expect, but the way we continue it also is to work with plants that show differential behavior and yet belong to the same line. Mm-hmm. So we explore differences not only between land races or between lines, but also within lands.
Mm-hmm. Okay, I see. I mean, it shows that.
I don't have more to say, because really it's very new and we are working on it. I don't have more to.
I mean, it strikes me that the individual dots there, they're all nicely packed to show that the variation across the field is not so great within a single genotype.
Well, see here, for example, this plant has low colonization and low yield, but in the same variety, for example, there is another plant that has higher colonization and higher yield.
Yeah.
So in this one, it seems there is a kind of correlation between increased diversity and increased yields. But this is not always the same. So this means that it is genetically controlled somehow. But in order to decipher this, we need to continue with precise experiments.
Yeah, it does strike me that there's an enormous amount of data that you're generating in these experiments.
That's true.
So to give us a bit of idea about the time scale for these experiments and collecting the data and getting all the roots stained and all that sort of stuff. How long does it take to do a complete analysis, if you will?
Well, you have to be really organized. Okay. Yeah. And the moment the roots are excavated, etc., and moved in the lab, you have to start working on that. But there are two different groups that are working. So the field group that has to be very well trained and know their work, and the lab group that has also to be very well trained. So usually within a season, we are able to finalize the plans for the season, the results of the season, and then have information about the next season. But it is a full-time job, right?
Yeah, absolutely. So let me ask just a final question. So you said at the moment it's 42 degrees Celsius in Cyprus. So how are the maize plants are obviously well adapted, C4 plants, they're well adapted. So do they grow okay in those temperatures still?
Well, they grow well. When it's we can irrigate them, of course, and we do irrigate them, but when the pollen gets destroyed, if the temperatures stay high for too long and it's the heat effect on the pollen, then we have problems also in the past where it's possible that the seed is not properly setting.
Yeah. I see. Okay, lovely. Well, I think we'll end it there. Thank you very much, Dionysia, for talking to us today. And as I said, the recording will be available soon on through the SEB app as well. yeah, we look forward to catching up with you soon, hopefully in Greece in a little while as well. So that'd be great.
Yeah, it's great that InDepth is going there. Thank you very much again for having me and for asking the questions so that I had the opportunity to say things that I forgot during my presentation.
Oh, my pleasure. No, it's interesting stuff. Thank you. Thanks very much, and we'll see you again soon. Thanks.
Okay, bye. Bye.
Bye-bye.